Good morning. Thank you very much, Neil, and thank you to the organisers for the opportunity, and thank you and congratulations to Samir on yet another wonderful fungal update meeting. What I'd like to do this morning is to take a look at what happens from the fungal perspective when spores are inhaled into the mammalian lung. And with the obvious exception of Candida species, the vast majority of human fungal pathogens enter their host via the respiratory route. So that's what we'll focus on in my talk. And I'm a founding member of the Manchester Fungal Infection Group. So what I thought I'd do for the first few minutes of my talk is to take you all up north to have a look around the labs. Video, please. And this is courtesy of a short video which was produced by our PhDs and postdocs in preparation for our official opening of the centre in late August 2015. So our mission at MFIG is to understand and reduce the burden of human fungal diseases, and our strategic vision is one of delivering healthcare benefits through the translation of basic biomedical sciences. And to deliver this, we've been working closely with clinicians and scientists at the National Aspergillosis Centre, headed by David Denning, the Mycology Reference Centre Manchester, headed by Malcolm Richardson, and which recently became an ECMM Centre of Excellence, and also the Manchester Collaborative Centre for Inflammation Research, which is headed by Tracy Hussle. So we have 4 key research themes. We're interested in the cell and molecular biology of the host pathogen interaction. We're interested in drug discovery and the molecular basis of drug resistance and target discovery, human susceptibility to infectious diseases. And in our inaugural 3 years of work, we've converted the initial £1.5 million investment from the university into a centre of more than 40 postdocs, PhD students and technical and support staff. And we've attracted more than £4 million worth of funding. 2/3 of that has come from the MRC. Amongst our major enabling technologies are high content, high resolution imaging of the host pathogen interaction, genome sequencing, so Paul Bowyer's been sequencing lots of human genomes from aspergillosis patients from the clinic. And our proximity to the clinic gives us not only an excellent source of primary human cells from at-risk patients in which to study basic infection mechanisms, but also very well characterised cohorts of patients in which we can test pilot interventative strategies and new diagnostics. We also have our own master's programme, which is headed up by Rena Richardson. And amongst our industrial collaborators are F2G, Evotec, Pfizer, and Blueberry Therapeutics. Okay, so in Manchester we spend an awful lot of time thinking about fungi and fungal pathogens. And there's one organism in particular that we focus on, and this is Aspergillus fumigatus, the major mold pathogen of humans. So fumigatus is a soil-dwelling mold which sporulates abundantly. And fumigator spores become airborne by thermal convection in huge numbers. They're often inhaled by human beings. And we know from the clinical cohorts at risk of these diseases that leukocytes are an important means of host defense against Aspergillus infections. And we also know that other types of immune deficits predispose to Aspergillus infection as well. And fumigatus is responsible for a spectrum of several different types of disease. So one of the questions that over the years I always seem to come back to is this one, and I'll throw it open to the audience this morning. There are around 200 known Aspergillus species on the planet. So why is it that amongst these, Aspergillus fumigatus predominates as a pathogen? So we can use our keypads now to vote on the answer to that question. Is it because A, the organism grows at 37 degrees, which is human body temperature? Is it due to the small spore size of Aspergillus fumigatus spores? Or has this organism evolved specific traits which favour pathogenicity? Or D, is it a combination of all of the above? And you can vote on that question now. Yes, and the answers are in, and we seem pretty much to agree on this. The most likely answer to the question is that it's a combination of all of these factors. Now, yes, Aspergillus fumigatus are small and buoyant, but remember the larger, much heavier spores of Aspergillus nigilans are also able to cause human disease, predominantly in patients who have a chronic granulomatous disorder. So although small spore size accounts for the frequency with which we see fumigatus infections, it can't entirely explain pathogenicity. Now, certainly fumigatus is a highly thermotolerant Aspergillus species. In this experiment, we cultured various Aspergilli in liquid culture across a range of different temperatures. And you'll see that in contrast to the lesser clinically relevant Aspergilli, fumigatus really excels in the 35 to 37 degree window of culture temperature. However, when we grow these species under identical culture conditions, but in the presence of human airway cells, we see a much clearer delineation of clinical relevance. And this indicates that amongst the Aspergilli, some species have evolved to be cytotoxic to human lung cells more than others. So our first insights into genes in the fumigatus genome which were switched on during mammalian infection came around 10 years ago. And this is a genome scale view of gene expression in a fungus infecting a leukopenic mouse host. So this work was done in collaboration with the J. Craig Venter Institute, and in this figure and in subsequent figures which depict gene expression, genes which are switched on are shown in red. So what we noticed in this experiment was that genes which were switched on during infection predominantly localized to the extremities of the chromosomes. These are the subtelomeric regions. And the black trace through the middle of each chromosome indicates the level of gene conservation amongst all sequenced Aspergillus species. So the results of this experiment were telling us that, well, what you'll notice is that this black trace peters out towards the ends of the chromosomes. So what the experiment was telling us was that fumigatus, during colonization of the host, is switching on genes which have recently evolved. And the other thing that we noticed in this experiment was that those genes tended to cluster together into physically co-localized groups. And we've recently revisited this work with Nancy Keller to identify which of those clusters are likely to provide the machinery for production of small molecule secondary metabolites in the host, and to define what this toxic cocktail of secondary metabolites could be during host infection. And this work was published last year in a paper we co-authored for the Emerging Fungal Threats edition that resulted from the Royal Society meeting last year. So we've looked in much greater detail now at the temporal expression of genes during germination and hyphal growth in laboratory culture, and also in several different animal models of disease. And one of the overall take-home messages we see from these experiments is that growth in laboratory culture bears little resemblance to growth in the mammalian host, and this pathogen has a specific pathogenic identity with respect to the genes it switches on when it's colonizing an animal. So we've become very interested in working out what the molecular basis of pathogenicity is in fumigatus, and we're taking a systems level approach to try to find the answer. So a systems level approach in fumigatus is a big job. Here comes the second question that I want to put to the audience. That should say question 2. Can you place these infectious organisms in order according to their genome size? So there's Aspergillus fumigatus, Mycobacterium tuberculosis, Plasmodium falciparum, and HIV-1. Can you place those in order according to the organism that has the highest number of genes? And you can vote on that now. Okay, so yes, answer C, this is the correct answer. Wow, well done, that's very, very good. 10 out of 10 for a Saturday morning. So actually this is an exercise which has to take place on a grand scale. So the fumigator's genome has over 9,000 genes. It's 1,000 times the size of the HIV-1 genome. And in Manchester, driven by Mike Bromley, there's been a drive to produce gene knockdown mutants which cover every gene in the fumigator's genome. So in a collaboration between Manchester and Axel Brackhager's group in Jena, this drive has now covered around a third of the fumigator's genome. But the thing is, if you want to take a house down quickly, you don't disassemble it brick by brick. You go for the important structural components that keep it standing. And the genomic analogy to that in fungi will be to take out transcriptional regulators, transcription factors. And these are the proteins that the fungus uses to direct these very important transcriptional signatures and gene expression in the host. The fumigator's genome encodes around 400 of these, and Mike and Paul Bromley at the University of Manchester have been developing a high throughput method for gene replacement in fumigators, and a number of years ago, before my move to Manchester, had completed construction of the transcription factor knockout library. So this is a collection of 401 transcription factor null mutants, and we've been working to develop an experimental pipeline which will allow us to measure the requirement for each one of these genes during pathogenicity. So as of yesterday, a team of 10 MPhil postdocs and PhD students spent all day yesterday harvesting 401 different Aspergillus fumigatus mutants, counting the spores, standardizing the inocular, and infecting those into 20 groups of mice. So we're looking at 20 pools of 20 different Aspergillus fumigatus mutants, and in the middle of next week we'll be able to cull those animals, harvest the lungs, extract the genomic DNA, and then we're going to use a high content, digital, high throughput quantitative PCR approach to measure fungal burden for every single mutant in every single mouse. And that should lead us quite quickly to the transcription factors which are driving pathogenicity in this organism, providing us with a genome scale census of pathogenicity factors. So once we've found transcription factors which are important in the whole animal model for pathogenicity, these are excellent tools which can lead us very quickly to a molecular understanding of pathogenicity in the organism. So this is an example of a mutant which lacks an important transcription factor called PAC-C, which helps the fungus recognize what the extracellular pH is. If we make a mutant which lacks PAC-C, we create a mutant which is unable to cause death in leukopenic mice. So we've constructed this mutant in several different genetic backgrounds now, and the result is consistent. And a very interesting phenotype that we've observed for this mutant is that the fungus loses its ability to invade lung tissue, and this is a landmark phenotype of Aspergillus fumigatus disease. And we've used this mutant as a tool to dissect this mechanism further. It's a relatively simple task, once you have a transcription factor knockdown, to go back into the animal model and ask which genes does that transcription factor regulate during infection. So this is the result of that type of an experiment for the PAC-C mutant. I'm showing you gene expression under lab culture in 2 mouse models for a wild-type fungus and then in the PAC-C mutant. So when we took this approach, what we found was that the non-invasive Aspergillus mutant was not producing gliotoxin, which is an immunotoxin. It was inappropriately remodeling the cell wall, but also it was misappropriately expressing a huge number of secreted proteins. So PAX-C is a transcription factor which regulates the expression of gene products which are exported from the cell in the host environment. And what we're looking at here is the first molecular picture of the tissue invasion phenotype, but also a list of genes, gene products, which could be important in terms of developing novel diagnostics and also perhaps novel biologics to prevent the tissue invasion phenotype. So we used the mutant as a tool to look at tissue invasion in more detail and set up some in vitro challenges in the lab using cultured human lung cells. And then we studied the interaction of the fungus with the cultured lung epithelium over a time series of growth from spores through swelling to beta-glucan exposure, germination, and then secreted product production, which occurs once the spore has germinated to produce hyphae. And in these experiments, we used a genetically modified fungus which expressed a red fluorescent protein, and that allowed us to visualize the fungus compared to the green backdrop, which is FIT-C concanavalin A labeled epithelial cells. We found a couple of significant events took place during this interaction. The first was that the lung epithelial cells internalized fumigator spores almost immediately upon contact. In this experiment, we used a differential fluorescence approach to distinguish between internalized fungal spores, which you will see showing up as red in these sections, and fungal spores which are stuck onto the outside of the epithelium and therefore remain accessible to a blue cell wall tropic dye. So what we're seeing here are internalized spores inside the epithelial cell against extracellular fungi which are stuck onto the outside of the epithelial surface and are now germinating and growing. So there are different fates for spores which end up inside the epithelial cells and are prevented from growing, and those which stick on the outside of the tissue and then grow on to produce secreted factors. So if we study this time course over a longer period of time and we look at cytotoxicity to human cells in the presence of the fungus, what we see is that challenge with live fungus during the initial host pathogen interaction leads to minimal damage compared to challenge with more mature hyphae or with culture filtrate from the fungus, which lead to almost immediate cell lysis and these big holes in the epithelial monolayer. And this experiment tells us that it's the secreted products from Aspergillus fumigatus which are really doing the damage in the lung setting. We've worked with Julian Naglick at King's College in London to identify which are the host signalling pathways which are being activated in response to the fungal secreted products. And we found that amongst the pathways we've analysed so far, one particular protein kinase mediated signal in the host cells is responsible for responding to secreted fungal factors. And if we block that host signal, we also block damage to the epithelium in a cytotoxicity assay. So actually, it's the host response to the fungal secreted product which is driving damage in our assays, and if we stop that host response, we can stop the damage. So we've used these same assays to challenge with different mutants from fumigatus. What we found is that our PAX-C mutant, the non-invasive strain, is unable to provoke this stimulus. We've also found that other gene products under PAX-C regulatory control, including gliotoxin production, including a broad domain regulator of protease expression, are also attenuated in this assay. And that tells us that fumigatus expresses more than one, possibly many more than one, separate gene products which are able to activate this pathway. And so with these assays set up and with a high throughput methodology at our disposal, we've now also worked with Mike to generate a library of mutants which lack individual secreted products. So we've gone after all 253 of the signal P proteins which are upregulated during infection by the wild-type fungus, and we're making gene knockouts for those. We're about halfway through that collection, and we've screened around half of those already in our cytotoxicity assay. So we're beginning now to get at the individual genes, the individual proteins which are driving damage during this disease. The other thing that we've been looking at is, other than the secreted products, what happens during that immediate interaction between the host and the fungal cell. So we've seen in our culture experiments that the spores are taken up by the epithelial cell. If we couple our differential fluorescence approach with a flow cytometric imaging approach, we can actually find individual epithelial cells interacting with individual fungal cells and ask ourselves, well, what's the fate of that interaction for the host? We've coupled the experiment with some markers of host cell death, so that one's an Nexin-5. If we see a red signal in this column, we know that the host-pathogen interaction has resulted in death for the host cell. We actually see very little of that during the early interaction, and overall what we found is that around 4% of sampled A549 cells, which internalise spores by 6 hours, are alive in our assay, and of those, after 8 hours, only 1% of the internalised fungal spores are still viable. So this appears to be an important mechanism of control for the host in terms of neutralising the fungal pathogen. And we're looking at whether or not this is relevant in humans. What I want to say towards the end of this talk is that it's important to think about stoichiometry when you do these experiments. So in mice, in vitro cultures, we very often challenge the host with very high numbers of spores, but in reality, the multiplicity of infection is probably much, much lower than that. So we know that the human lung has around 700 million alveoli, and that equates to around 2 billion alveolar epithelial cells. But we also know from air sampling studies that humans inhale probably several hundred spores on a daily basis. So actually, that's around 1 fungal spore to 10 to the 7 human epithelial cells. If the cells in our lung are doing anywhere near as good a job as the ones that we've been studying in vitro, the epithelial cells in the alveolar portion of our lungs are probably quite important in terms of controlling fungal burden. And all the more so if you think about the lower extremes of the respiratory tract, where the mucociliary clearance mechanisms are absent. So what we're doing now is working through our at-risk clinical cohorts through the clinic in Manchester to see whether or not we see in primary human cells any deficiencies in spore uptake in groups of patients who are predisposed to infection. So in summary, I've told you that Fumigatus is a transcriptionally versatile organism, particularly under host-imposed stress, that it coordinately regulates multiple secreted factors and those govern epithelial damage, that in contrast to internalised spores, it's those which germinate extracellularly which activate host signalling, and that's in contrast to what Liz described for crypto in the first talk, and that leads to epithelial damage. And also the uptake of Fumigatus spores occurs in vitro. We've also seen it in whole animal experiments, but I didn't show the data. And this may provide a fungicidal defense mechanism, possibly providing immunotolerant disposal of inhaled spores before they then go on to germinate and become problematic for the host and the immune system. I want to finish up with a final question before the end of the talk, and this one's just for a bit of fun. Can you have a look at these famous scientists and tell me which one is the odd one out? Okay, so you can start that now. Please vote. Split decision. Interesting. Okay, so let's have a look and see who these are. The answer is C, so I suppose the majority got that correct. But what we're looking at here are winners of the Nobel Prize for Physiology or Medicine, which over the years has been delivered to scientists working on antiviral, antibacterial and antiparasitic drugs. At the moment, nobody's secured the accolade of a Nobel Prize for an antifungal drug discovery. But this individual, I don't know whether you recognised him, was David Denning, and I'm not suggesting that he's the next candidate, more that he's yet another accolade that the fungal community has yet to secure, so watch this space. So with that, I'd like to thank everybody at the Manchester Fungal Infection Group, particularly David Denning, for having the vision to drive the establishment of this excellent group, especially Mike Bromley, who's been an amazing collaborator, and they're very exciting times for us in Manchester with the work that we're doing at the moment, to Neil for the Wellcome Trust support. I also want to note Joy, who's funded by this grant and has done a lot of the epithelial work with Julian at King's, and also the Chelsea and Westminster Health Charity. So 12 years ago, at the first one of these meetings, I met a trustee from this charity who began to fund the epithelial studies in my lab. And since that first meeting 12 years ago, I've had many tens of thousands of pounds of funding from the charity as a direct result of speaking at this meeting. So thank you, Samir, for that. And thank you all very much for your attention.